Hello, BISC132. This is the beginning of recorded lecture 5.2, continuing on with the immune system. So last time we had started this chapter by talking about the innate immune system. We're still under that category. And we're still talking about leukocytes, these white blood cells. So before I can talk about the next type of leukocyte, we need a little bit of backstory. So all cells in the body, not just leukocytes, not the immune system, all your cells, your muscle cells, your epithelial cells, all the stuff, all cells in the body display pieces of proteins they have been making on a major histocompatibility complex at MHC class 1. That's a Roman numeral 1, MHC class 1 molecules. So again, any healthy cell, any cell in the body has this MHC1 on its surface, and it puts little fragments of proteins on there, basically to just show the outside world, to show all the rest of the cells, hey, look at what I've been working on. This is what I've been making. Little pieces of any protein, all the normal proteins, any protein that a cell makes. It's going to put some sample of that in the MHC class 1. Now, if a cell becomes infected with a virus, or if it becomes cancerous or on its way to cancer, that cell could have problems in making its MHC class 1. Either it will display no MHC class 1 at all, as we're seeing here, or it could display a messed up version of the MHC1 that just doesn't look normal. It's got mutations in it. It's weird and abnormal in some way. So we collectively call these virus-infected or cancer cells altered self cells. So cells that are virus-infected or cancerous, altered self cells, might display no or abnormal MHC class 1. So they might not do it at all, or they might have a messed up version of MHC class 1. So, okay, again, this is all just backstory. This is how the cells in your body work. This is what they do. Now, something we saw last time, earlier in this chapter, was that the cells of your immune system, these leukocytes, deal with problems by eating them, phagocytosis. This works for a virus, this works for a bacterium, but this is not going to work for one of your own cells that's gone rogue. It's just too big. You can't eat this cell to destroy it because it's pretty much the same size as the phagocytic leukocyte itself. So this is where we bring in a type of leukocyte called a natural killer cell. So, very, very grim name, or because we need more abbreviations, an NK cell, natural killer cell. They are looking for MHC class 1. And if a natural killer cell sees a normal healthy cell displaying its MHC class 1, doesn't matter what protein is on the MHC1, but if it's got normal MHC1, it gets the green light, it's fine. However, if you have one of these abnormal self cells that is displaying no MHC1, or, not pictured here, but you can imagine an abnormal version of MHC1, that natural killer cell is going to, again, not do phagocytosis. It's going to induce something called apoptosis in this altered self cell. So natural killer cells, these are leukocytes, part of the innate immune system. Natural killer cells cause apoptosis in altered self cells. It's spelled apoptosis. It's supposed to be apoptosis. We talked about this in an earlier chapter in BISC130, but it's in the key terms again for this chapter. Apoptosis is programmed cell death. Every cell in your body has the instructions for destroying itself, and this looks like a dramatic Death Star explosion. It's actually a very orderly, controlled process of this altered self cell, either cancerous or virus infected, destroying itself so it doesn't destroy the rest of the body. So this is that's what natural killer cells do. And again, this is part of the innate immune system. This is not going after a specific virus. It's just any cell that happens to not be showing its MHC1 or abnormal MHC1 is going to be targeted by natural killer cells. Okay, now there's one last thing in the innate immune system, and it is something called complement, or the complement system. And oh man, this is complicated, but we're lucky for you, we're not going to memorize these pathways. But needless to say, there are a couple of different ways to start this, and it involves a protein being cut in two, and one of those fragments cuts another protein in two, and that protein goes on to recruit other proteins, and you get more alphabet soup of complement 5B and 6 and 7, all this stuff. Don't memorize all this, but I did want to show this so you appreciate how complicated this is. The short version of complement is that this is a series of proteins, a system of proteins in the bloodstream that ultimately lead to lysis of a pathogen. They lead to this formation of an attack complex in the pore of the pathogen, and it starts leaking stuff in and out, and the pathogen dies because it has these pores formed in its surface. So this is all I want you to take away from complement. This system uses a complex array of proteins in the bloodstream that ultimately lead to lysis of the pathogen. And again, this is not super specific for a pathogen. That's why this is still under the innate immune system heading. We're going to bring up this later. The reason why it's called complement is because this pathway actually works with part of the adaptive immune system. So we'll actually come back to complement once we understand what's going on with all this stuff up here. So complement, because it kind of brings the two systems together. But this is all we're saying about complement for now. Okay, that's it for the innate immune system. On to the adaptive immune response. So the innate immune system was kind of generic, but was really fast. The adaptive immune system is very strong against very specific threats, but it has a longer lead time. It takes longer to really kick in. Okay, so the adaptive immune system is going to involve several different types of leukocytes, more white blood cells. Let's start by talking about B cells. If you're curious, they're called B cells because they complete their maturation in the bone marrow, hence B, but whatever. B cells are responsible for what's called the humoral immune response. B cells create antibodies. This is a very important concept in immunology, and I'll show you what an antibody looks like before reading the key terms definition. An antibody is a protein. Actually, it's several different polypeptide chains that are connected together. That's a quaternary structure, if you remember that from BISC132. But yeah, this is a large protein complex that has a strange and unique Y shape to it. Most proteins don't look like this, but yeah, it's a Y-shaped protein. The key terms define antibodies as a protein that is produced by B cells, also known as an immunoglobulin. So yep, we're getting even more fun by having multiple names for these things, but immunoglobulin antibody, same thing. And we'll often see these called ABs, just an abbreviation for antibodies. So this is a protein. What does it actually do? Antibodies bind to antigens. So this is an antigen. What is this blue blob meant to be? It's very generic for a reason, because an antigen can be a lot of different things. An antigen can be a protein, a carbohydrate, a small molecule. So it's just, the definition here is a little circular. The key terms definition of an antigen is a substance that an antibody binds to. So if an antibody can bind to it, it's an antigen. And again, this could be a protein, this could be a carbohydrate, this could be part of a lipid, this could be part of a small molecule. Just if an antibody binds to it, it's called an antigen. Now, to be a little bit more specific, an antibody is not going to bind to the entire antigen all at once. In fact, if we go back to the antibody here, here's the antigen binding site, this little region here at the tip of the Y. There are 2 of them per antibody. So it's not going to bind the entire antigen, it's just going to bind to a specific structural part of the antigen. That term is the epitope. So It is true to say an antibody binds to an antigen. It is also true to say an antibody binds to an epitope of an antigen. Here's crudely combining these two together. This antigen binding site of the antibody is going to interact with this specific structural region of the antigen, the epitope. The key terms definition of an epitope, a very specific structural region of an antigen. So here's a summary of this. Antibodies have this distinctive Y shape, the two tips of the Y there, two antigen binding sites per antibody, and these bind to the epitopes of antigens. Here's another figure showing this. There's antibody, here's an antigen, and yeah, it's not binding to this corner or this side. It's the specific region or epitope of the antigen that the antibody is binding to. Now, a very important part about this, and part of this figure is illustrating this, antibodies are very specific for the epitope that they bind to. So this is showing there are a bunch of antigens that exist. Just any protein or anything that has a molecular structure is potentially an antigen. But in this example, this antibody binds to this antigen, the A, not the B, not the C, not the D, not the E. It's a specific interaction between this antibody and this antigen. Now, you have a lot of B cells in your body because you want to be able to respond to a bunch of different antigens. You don't want to just have antibodies that bind to one specific thing. You want to have a lot of antibodies in your repertoire to be able to deal with any possible threat. So each B cell that you have, because remember we're talking about B cells that make these antibodies, each B cell produces its own unique antibody with unique epitope binding capabilities. So, this antibody came from a certain B cell. That B cell always makes this antibody, and this antibody binds to this A antigen. But maybe you've got another B cell in your body that makes a slightly different antibody that can bind to this C antigen, and so on. You've got millions of different B cells in your body, and each one is different and unique, which might make you wonder, how is this possible? You've got millions of different B cells. Each one has its own antibody. These are proteins. Proteins come from genes. What do you got? Millions of different antibody genes? That seems incredibly wasteful. How can you fit all those genes into a chromosome? Well, we don't. There's a workaround. The way that we get millions of different B cells is by genetic rearrangement. And this is crazy because on a conceptual standpoint, it is very dangerous for a cell to mess with its own DNA. That's how you get mutations. That's how you get cancer. That can cause problems. So B cells do this, but under very controlled circumstances. So they have multiple different versions of this part of the antibody protein and multiple different versions of this part. And again, skipping a lot of details here because it's beyond the scope of an intro course. They will cut away parts of the chromosome, whittle things down until they've created their own unique antibody gene. Every single B cell in the body is going through this process of random gene rearrangement in order to create a unique, randomly generated antibody that, binds to some unique epitope of an antigen out there. So again, each B cell makes its own unique antibody. This is achieved by randomly rearranging the DNA for the antibody gene in maturing B cells. So, as I've been saying, this means there are millions of different B cells in the body, so there are millions of different antibodies. This allows the immune system to respond to many possible antigens. Again, this is the adaptive immune system. This isn't a generic pattern recognition receptor that, recognizes some common danger pattern. Anything the world can throw at you, any antigen that you could possibly encounter, could have a corresponding antibody because you have so many of these that are available. Now, one thing to ask is, this is all well and cool, but where are these antibodies in the B cell, and how do they actually work? How does this help you? So let's answer that first question first. Antibodies are present in a couple of different places. Antibodies are present on the surface of B cells. So let's look at this figure here. There's a B cell. There's an antibody. Actually, it's not just an antibody. It is this B cell's specific antibody. Again, nothing else is like it. It's specific antibody. And here it is binding to an antigen present on a bacterium. Now, this is not something that happens all the time. Because B cells are making their antibodies randomly, most B cells are useless. This is sad to think about, but I mean, imagine if you had a key and you were trying to open, a lock, and you just randomly built your key. Most of those keys are going to be useless. They're not going to open any lock. So most B cells never get to experience this. But if a B cell actually encounters the one thing that it is capable of binding to, that's game on for that B cell. The one thing that you are able to fight against is now actually present in the body. Well, before it can go time, it needs a bit of an activation, a counter signal. This is where helper T cells come in. We're kind of skipping ahead because as soon as we're done talking about B cells and antibodies, we'll get to T cells. So I'm kind of skipping ahead, but go back and revisit this once we've done everything because it's all connected. So once the antibody on a B cell has found its antigen, a helper T cell is going to recognize this. It's got its own MHC class 2 and T cell receptor that is going to recognize this presented antigen, and that's going to sort of give the green light for this B cell to have its time to shine, its go time. Once it's been activated by this helper T cell, this specific B cell with this specific antibody is going to make a bunch of clones of itself. All of those clones have the exact same antibody and antibody gene, and all those clones are going to start spewing out antibodies like crazy. They're called plasma cells or plasma B cells. So again, it's action time because this doesn't happen for most B cells. So antibodies are present on the surface of B cells. If they bind their antigen, they present it on their MHC2. So again, MHC class 1 was something that every single cell in the body has to say, hey, this is what I've been working on. MHC class 2 is only present on certain leukocytes. And here's this B cell, showing the world, hey, I've encountered this antigen. Is this something bad? Is this something important? And it takes the helper T cell recognizing that to say, yes, it is. Go forth, fight. So helper T cells recognize this antigen on the MHC2 and cause that particular B cell to replicate, meaning clone itself, and become plasma B cells. Plasma B cells secrete tons and tons of antibodies. Now, the other question that I brought up. So what? we have these antibodies. They are, specific to a threat. I mean, this is a bacterium. This is a bad thing. How does this help us at all? Well, there are three ways. One is by neutralization. So, as it turns out, a virus that is coated with antibodies can't do its thing. I mean, if you remember our chapter on viruses long ago, they're supposed to bind to the surface of their host cell, protein-protein interactions, facilitating that. If this virus particle is coated with antibodies, it's not going to be able to do that. And toxins, like this diphtheria toxin, similarly, if they're coated with antibodies, they're not going to be able to do whatever it is they're supposed to do. So this is one way that antibodies help us. They neutralize viruses or toxins. Another way that they help us with against bacteria is by something called opsinization. So here's a macrophage again. Remember, these are phagocytic leukocytes. We brought these up when we were talking about the innate immune system. They're brought to the site of wound injury, and they do phagocytosis, but they're not very smart. So at a wound site, they know that they're supposed to engulf stuff around them, but in any other context, they don't necessarily know what's good and what's bad, what you're supposed to phagocytize and what you're not supposed to phagocytize. But if something is coated with antibodies, that tells the macrophage, okay, this thing is definitely no good. I'm going to do phagocytosis on it and destroy it. So this process sort of tags pathogens to be phagocytized, sort of pulling in the innate immune system. I said everything's connecting this to adaptive immune system, but you need antibodies to do this, to tag these pathogens. And finally, antibodies activate complement. So antibodies can also tag pathogens to be lysed by complement. So yeah, here they are binding to this pathogen. This just shows 4 pores formed by complement. If we could go back to this again, we could see it's part of that complex array of chemical reactions. Yep, there's the antibody, antigen, and this interaction, again, without getting into the details, activates complement. So these are the 3 very important things that antibodies do. And so B cells, producing these antibodies are definitely something that helps us fight against these pathogens. Now, one last thing about B cells, or one last thing about antibodies. I had said several times earlier that this specificity is very high, that this antibody is going to bind to A, but it's not going to bind to D or E or B or whatever. That's true up until a point. So it is possible to have 2 antigens, or I guess 2 epitopes that are very similar to one another. And if you have 2 epitopes that are very structurally similar, sometimes you can have a single antibody that binds to these 2 different epitopes. They have to be very similar, but one antibody, 2 epitopes. This is called cross-reactivity. And this can be good or this can be bad. So sometimes different antigens can have the same or very similar epitopes, resulting in cross-reactivity. The key terms define cross-reactivity as binding of an antibody to an epitope corresponding to an antigen that is different from the one the antibody was raised against. So, okay, is basically what I already said, but here we are. So what does this result in? Well, this can be good if your antibody against a specific pathogen grants protection against another similar pathogen. Maybe you have a slightly different type of flu virus that happens to have a very similar epitope. Hey, your reaction against that virus is valid against this one too. So cross-reactivity can benefit. Or it can be bad when these antibodies are cross-reacting with some pathogen that they were created to fight, and something very, very similar looking that's in the normal human body. We'll actually get into that a little bit later in the chapter when we talk about autoimmunity, but either way, know the definition of cross-reactivity, a single antibody binding to different things. So that's it for B cells. On to another leukocyte in the adaptive immune system, T cells. Again, if you're curious, they're called T cells because they complete their maturation in the thymus. It's part of the lymphatic system. Not getting into that, but anyway, T cells. T cells are responsible for what's called cell-mediated immune response. T cells do not have antibodies. Please don't get these mixed up. Only B cells and plasma B cells have antibodies. T cells have something kind of like an antibody, but different in a few ways. T cells have something called a T cell receptor. So let's compare and contrast this with an antibody. Well, like an antibody, T cell receptors have an antigen binding site. Unlike antibodies, there is only one antigen binding site, and unlike antibodies, T cell receptors are always embedded in the plasma membrane. They are never, floating around and binding to stuff outside of a cell. So the similarities, with antibodies, keep going. If we look at this antigen binding site, it's exactly the same as it was with the antigen binding site of an antibody. So these T cell receptors are similar to antibodies in that they bind a specific epitope of an antigen. They're unique to each T cell. You got millions of different T cells, so you have millions of different T cell receptors, and it's the same thing, genetic rearrangement during development leading to all these different T cell receptors. But again, not the same thing, only one antigen binding site per receptor, and they never leave the membrane. They're also different in what they do and how they're used. So there are three types of T cells. The first one I want to look at is helper T cells. Oh, should sound familiar. Yeah, we already talked about this when we were talking about activation of a B cell into a plasma B cell. So this is easy. The job of helper T cells is to activate an immune response when their T cell receptor binds to an antigen displayed by an immune cell. That's exactly what we saw here. So here's the T cell receptor on this helper T cell. It's recognizing this antigen being displayed by the B cell that encountered it, and yep, it helps the immune system go forward. So that one's simple enough. Another type of T cell is the regulatory T cell. They, instead of activating an immune response, they suppress an immune response in response to harmless antigens, stuff like food. So again, an antigen is just anything, any structural thing, any shape at all. And so a lot of antigens that you come across, most of them are completely harmless. You're encountering them in your food, and you don't want to have an immune response to all this stuff. So the Tregs or regulatory T cells are a big part of that. It's called immune tolerance, defined in the key terms as the acquired ability to prevent an unnecessary or harmful immune response to a detected foreign body known to not cause disease, or to self-antigens. You also want to suppress an immune response to, fight yourself. More on that later in the chapter. So the third type of T cell is called the cytotoxic T cell. So to understand the cytotoxic T cell, we kind of got to go back to this. So remember, MHC class 1 wasn't just a protein on the surface of all cells. MHC class 1 actually carries a little bit of a specific protein that these cells have been working on. And natural killer cells, these were part of the innate immune system because they didn't care what protein was on the surface here. They weren't specific for whatever protein was on MHC1. They just cared about whether there was MHC1 at all in the first place. Cytotoxic T cells, however, are specific for the epitope on that MHC class 1. So, okay, let's orient ourselves here. Here's the T cell. There's the T cell receptor. Here's the class 1 MHC on this, it says infected cell in this case. And the little black triangle here is supposed to be the actual protein. So this could be a viral protein that this cell has been making because it's infected with a virus. Or it can be some screwed up cancer version of a normal protein. But either way, it is not a normal antigen. It's not a normal epitope. And this cytotoxic T cell has a T cell receptor for that particular antigen. So the cytotoxic T cell causes, just like the natural killer cell did, apoptosis in this infected or cancerous, in this altered self cell. But again, the thing to appreciate is the difference. Natural killer cells were just looking for, does it have no MHC1? Does it have abnormal MHC1? The cytotoxic Ts are using their T cell receptors to look for very specific foreign antigens presented on the MHC class 1. But the end result is the same. Cytotoxic Ts cause apoptosis in altered self cells. If their T cell receptor reacts with an antigen presented on the cells, MHC class 1. Okay, so that's it for T cells. My next statement is going to apply to B and T cells. So we're talking about both of these leukocytes now. Activated B or T cells can become memory B cells or memory T T cells. So these memory cells are kind of like veterans. There are a lot of military analogies that you could try to draw when you think about the immune system and, fighting off foreign pathogens or things that are causing harm to the body or whatever. And after their battle, some of these B or T cells that participated in that battle will become memory cells. Memory cells are incredibly long-lived. We're talking years, sometimes decades. That's a much longer lifespan than a normal B or T cell. And what these memory cells do is they help fight against that pathogen if it comes around a second time. So I think the best way to see this is in graph form. So this is showing, this is just looking at memory B cells, but you would see a similar thing in memory T cells because this is looking at concentration of antibody. You're exposed to some pathogen. There's a lag time. Again, the innate immune system is fast. The adaptive immune system takes a while to get going. You have your primary immune response. You have a certain number of antibodies. They have a certain affinity. The infection is gone. You survived. Hooray! And you have some memory cells. Once you're exposed to that same pathogen a second time, those memory cells, these veterans, are way better fighters than they were when they were still green, when they were still new to this. This is called the secondary immune response, where these memory cells kick into action faster. I mean, look at the exposure to peak time as opposed to exposure to peak time. They kick in faster and they have a much higher, more robust response than the initial immune response. Oftentimes, this secondary immune response can be so strong and fast, it prevents you from even feeling sick. You've been exposed to some pathogen, but your body fights it off before you can have any symptoms of that infection at all. So, these veterans do a great job of protecting you. So these memory cells provide rapid, strong response to their antigens on second exposure. This is called the secondary immune response, and this is going to be involved when we talk about vaccines and how vaccines work. But that's going to be later in the chapter. So one other thing to bring up when we're talking about B and T cells, and that is the sort of cross-reactivity or improper reactivity. Because B and T cells are creating their antibodies and T cell receptors respectively with some amount of randomness. This is for antibodies, but the same sort of random rearrangement is happening for T cell receptors. Because there's randomness involved, it is very possible to create a B cell or a T cell whose antibody or T cell receptor reacts to antigens in the normal human body. It's gonna happen. You can get B or T cells whose antibodies or T cell receptors react to normal, harmless stuff that's just part of the normal human body. These cells are supposed to be killed during development. All of these have, very lengthy, complex developmental cycles, like boot camp, I suppose, if we're continuing the military analogies. And if they have a tendency to shoot their teammates or whatever you want to do the analogy for, they're supposed to be taken care of, destroyed during development. So these self-reacting B or T cells are killed during development, or at least they're supposed to be. Failure to do this results in hypersensitivities and or autoimmunity. So this plays a role in autoimmune diseases and in allergies. So the definition of hypersensitivities, just reading from the key terms, a spectrum of maladaptive immune responses. So that means this includes a bunch of ways of the immune system behaving badly. A spectrum of maladaptive immune responses toward harmless foreign particles or self-antigens occurs after tissue sensitization and includes immediate type and delayed type and autoimmunity. So this is a big umbrella term. It includes a bunch of, improper reactions to things, including allergies. So don't sweat the details here, but Here's a B cell reacting to ragweed pollen, completely harmless, producing a whole bunch of this immunoglobulin IgE. We didn't even talk about the different types of antibodies. Don't worry about it. But yeah, and here is the second exposure binding of that antigen to the IgE, again causing all these horrible symptoms. And it all started with this improper reaction to ragweed pollen, something completely harmless. So hypersensitivity, again, is kind of like an umbrella term that includes a lot of things that your body does that it's not supposed to do, including allergies. This also includes autoimmunity. So autoimmunity is a type of hypersensitivity. Reading from the key terms, autoimmunity is a type of hypersensitivity to self-antigens. So this is specifically, don't memorize this, I say that a lot, but this is these are some common autoimmune diseases where your body attacks some of the insulating cells on your neurons, resulting in multiple sclerosis, or attacks cells within the gut, or attacks the kidneys, or attacks cells within the pancreas. Actually, we'll swing back to type 1 diabetes when we get to our chapter on the endocrine system. But all of these are examples of the body attacking part of itself because of a failure of these self-reactive B or T cells to destroy themselves during development. Okay, there's one last section on the immune system. I gotta talk about vaccines, but this is typically where I run out of time in the in-person lecture. I don't want to have to rush through vaccines. It's a very important topic in my opinion. So this is where I'm going to cut things off for here. This is the end of recorded lecture 5.2.